Hello students, now we are going to discuss the plant tissues in detail. Those tissues which basically make the plant body. You know what a plant body is made up of? It is made up of three main organs: shoot, leaves and root. So we are going to talk what these systems are made up of, what are the tissues which are behind the composition of these organs. So on the basis of different classifying features, we have two types of plant tissues. First one being the meristematic and the second one is the permanent tissue. Meristematic tissue is that tissue which gives rise to the next tissues which happen to become the permanent tissues. So first of all, let's see what these meristematic tissues are all about. We have further classification of meristematic tissues on two bases. There are two bases of classification. First being the origin, that means from where they have originated. On basis of that, we have three types of meristems. One is promeristem. Pro means first, as you know, so this is the first formed meristem. Then is the primary meristem and the third one is the secondary meristem. These three types of meristems are classified on the basis of origin. That means how they have originated when the plant was initially in its seed constitution. The promeristem comes first, then comes the primary meristem and then comes the secondary meristem, from which the entire plant body would get derived from. On the basis of position, then there are three types of meristem again. Apical meristem, which is situated at the apex, intercalary meristem, which is found in the grasses, and the lateral meristem, which is responsible for increasing the girth of a plant. So there are in total 6 types of meristems, as you can see, but please don't confuse it that this is a different type, this is a different type and these types are totally different. It is a possibility that the apical meristem would have originated from the promeristem only. The apical meristem would have primary meristem, the intercalary meristem would have these meristems. So basically the basis of classification is different, that's why we are having different types. Otherwise, if we see in a plant, if we see a plant's diagram, that is, it is having a root system and a shoot system. Root system is below the ground and shoot system above the ground. So basically then we have these three types of meristems which are present. First being the apical meristem. As the name suggests, apical meristem is going to be present at the apices, or we can say the apex. The shoot apex, that would be the tip from where the new leaves would originate, would have the shoot apical meristem, and from where the root is elongating positively geotropic fashion, that would be the root apical meristem. Inside this apical meristem, initially there would be presence of promeristem. Please pay attention. Inside the apices, there would be promeristem initially, which would further divide to three types of meristems or the regions according to older types of classification of meristem. First being the dermatogen. You know the term dermato, it is used for skin specialists. You might have heard dermatologist. So it is dermatogen, it is signifying the skin. That means it is going to make the epidermis of the plant. Then comes periblem and then comes plerome. Dermatogen, once the skin has been formed in the apex from the promeristem, periblem would form the cortex and plerome would form the procambium, from where the vascular tissue would originate. Vascular tissue, you must remember, it is the xylem and the phloem in plant which is responsible for carrying out the activity of conduction and transport of sap. So we have promeristem differentiating into dermatogen, periblem and plerome, which further differentiates into the epidermis. Periblem differentiates into the cortical region and plerome develops the vascular tissue, and all these are present in the apical meristem, and these apical meristem are situated at the apices of both root and shoot inside the plant. Now we have intercalary meristem, which is exclusive to the grasses. You must have seen that when a lawn grass is mowed, it comes after 2-3 days. If you pluck grass, it grows. If the grass is grazed, then again it grows. So the meristem which is responsible for its regrowth at such a pace is the intercalary meristem. So only point that is to be remembered in intercalary meristem is that this type of meristem is present in the leaves of grasses which are monocots. So one point of remembering is that it is exclusive to monocot plants. I hope that you know the difference between monocots and dicots. If not, for once just remember dicot plants are those which are bulky plants. They are having girth, they have stem, trunk, the trunk basically, not the stem, leaves, as in case of mango. And monocots are simply grasses, wheat, rice, the grasses that grow like weeds, they are monocots, the simple differentiation. And the last one on the basis of position is the lateral meristem. Now as I told you that a typical plant is like this. Since childhood we have been drawing a trunk, basically a trunk, and it starts from a very small sapling which is not having trunk in the beginning. Its transition from this stage to this stage where a girthful trunk has been formed is caused by the lateral meristem. And as you must be knowing, right now I told you the difference between monocot and dicot. That means this lateral meristem, which is providing girth through the plant, would be exclusively present in the dicots. So another point of remembering is that the lateral meristem is known as secondary meristem because it develops in the later stages of plant growth. So lateral meristem happens to be the secondary meristem, and the meristem which develops initially, from which the secondary meristem would derive, would be known as primary meristem. So these three terms which I wrote over here happen to be primary meristem because all the plant tissues which would be termed under permanent plant tissues would originate from primary meristem. I hope you are able to make a clear-cut distinction between the basis of classification and how this meristem would be located inside the plant cell. Please make sure that these are not 6 types. These are bases on which the classification is made, and bases being different in both the cases, we have 3 types. They would be located in the same position only. As I told you in the case of apical meristem has promeristem, which further differentiates into these 3 types, which are a type of primary meristem. Then there is lateral meristem, which is located in lateral positions of the plant, and this lateral meristem happens to be the secondary meristem, which is derived from primary meristem in later stages of plant growth. All these tissues and the types that we have studied under it come under the type meristematic. Meristematic cells or the tissue, whenever we talk about the meristematic tissue, has those cells which have the ability to give rise to new cells. They do not leave their ability to divide. That is why a meristematic tissue can give rise to entire plant and still not lose its ability to divide. This is the entire classification of meristematic tissue. Now, as I told you in the beginning, there are 2 types of plant cells, first being meristematic and the second one is permanent. So, in this lesson, we are going to take into discussion the simple permanent tissue. Now, when I talk about permanent tissue, the permanent tissue does not have the ability to divide as in the case of meristematic tissue. There is one point of remembering that you need to pay attention to, that this tissue has undergone differentiation. You might have heard of this term, or in case you haven't or you don't have a better idea, please pay attention. Meristematic cell gives rise to a cell which is of its own type. Later on, this meristematic cell's precursor or successor, it gives rise to the cell which has its specific functions allotted to it. It performs the specific functions, and it would not give rise to a cell which would be meristematic again. It would give rise to the cells after division to those cells which would be similar. That means cell division would work on it, but it would not be meristematic division. That means these cells have undergone differentiation. It is just like in the beginning you have made some few slots. After that you give work to those slots. That means they have been given a work, so they have gone differentiated from the meristematic tissue. That is why we call them differentiated tissue. That means they are no more like meristematic tissue. They have a specific function now which they would be performing, and they have been differentiated into a special class of cells. Now those class of cells further form the tissues, and if all the cells inside the tissue are similar, then that tissue happens to be simple permanent tissue. With this I mean to say that supposedly this is a tissue area. Supposedly all the tissues inside it would be similar. They would be similar in shape, they would have similar function, they would have similar attributes to them. There would be no distinction in the structure of cell which form the tissue. Supposedly these are isobilateral cells. Over here I am making hexagonal cells. So all of them would be hexagonal. So such a tissue arrangement would be known as simple permanent tissue. And when I talk about meristematic, on the contrary, the similar cell, small size cell, full of the cytosol, very much actively dividing, would always give rise to similar cells. They do not have any differentiation, they do not have any work, but these cells, they have been differentiated, they would be similar, so they would be termed under simple permanent tissue. The simple permanent tissue is further of three types. First is parenchyma, second is collenchyma, third is sclerenchyma. Please pay attention. Every part of the plant, whether it is a leaf, whether it is a stem, whether it is root, is going to be formed of these tissues in any case. In this, you need to remember that this type of tissue is again an exclusive character of dicot plants. Dicot plants, these one which increase in girth. Example, mango tree. So the collenchyma tissue would not be found in grasses. First thing to be remembered. Next thing that you need to remember is that every part of the plant, whether you are eating a fruit, you are seeing a flower, would be made of these tissues, which would be formed from the meristematic tissue in the beginning. Now let us see what this parenchyma is all about. Parenchyma tissue is formed of simple cells, okay, because it is a simple tissue, it is going to have all similar types of cells. So pay attention. If we study one cell, we get to know what other cells would be made up of and what the tissue composition would be. The cells are isobilateral, or they have similar diameter. That means they would be somewhat circular. So now this is circular, so the other one would also be circular, somewhat spherical we must say, because the cells are in 3 dimension. They have intercellular spaces in between them. They are isodiametric. They have intercellular spaces in between them. There is lot of protoplasm in between them, and their function that they perform is storage and secretion. So if we see a leaf, maximum of the part of leaf would be formed up of parenchyma. This parenchyma may have chlorophyll inside it. If it is present in the leaf, it would be termed as mesophyll. If it is present in the stem, it would be simple cortical parenchyma. So parenchyma forms the main bulk of the plant. It is the tissue which is responsible for storage, secretion and photosynthesis basically. If it performs photosynthesis, it is known as sclerenchyma. If it is having air vesicles inside it, it is known as parenchyma, and this parenchyma is the main bulk. Now we come to collenchyma. Collenchyma tissue will not be present in monocots, please remember it. It would be present in the hypodermal region of a dicot stem. These tissues are, unlike parenchyma, they are somewhat hexagonal in shape, okay, and unlike parenchyma, which has cellulose cell wall around it. They too have cellulose cell wall. They are closely packed, but a peculiar feature which is present inside the cells of Collenchyma is that they have angular thickenings. Angular thickenings means there would be some deposition of primary nature at these places. These angular thickenings have deposition of hemicellulose and other derived compounds which provide it a hard structure. So this Collenchyma basically is present inside the stem of a dicot plant, which gives it extra stability and mechanical strength. Then we come to Sclerenchyma. You might have eaten chiku, you get some grainy taste inside it. That is Sclerenchyma, what I am trying to tell you. You might have eaten a walnut, you need to break that wood. That wood is not wood, please remember, because we will get to know what wood is. That is the sclerenchymatous tissue. Sclerenchyma is further of two types based on the type of cells present in it. One is fibers and other is the sclereids. Now, you must remember that only this part and these are living inside the plant. This tissue is dead. It is not living. By dead, I don't mean that it must be roaming like anything. The dead means that it would be devoid of protoplasm. There would be only cellular composition, but protoplasm wouldn't be there. No metabolism would be carried out. So we have fibers and sclereids, and this classification is made on the basis of lumen. Supposedly this is a sclereid which is having its outer boundary, and they have a narrow lumen inside them. So they would be classified as fibers. In terms of sclereid, the wall lumen gets much shorter. There is a thick wall which is present. The thickening has been formed by the deposition of lignin and pectin, which makes it hard. That is why the walnut that you have, you need to crack it with teeth or some nut cracker. That hardness is being provided by this Sclerenchyma, dead Sclerenchymatous tissue, which is going to be present everywhere inside the plant. And all these three types are simple tissues which have same types of cells inside them. Then we have complex tissues, which is xylem and phloem, which would be covered in the next lesson. Thank you.